Transcription and the regulation of transcription. All right, we're going to talk about gene expression with regard to, what is cis, what are promoters, what are enhancers, how do they interact with transcription factors, what is the transcription mechanism, what about introns and splicing and post-transcriptional modifications, and then regulation, mostly focusing on the lac operon. Why is a muscle cell different than a liver cell? They have the same 3 billion nucleotide code, or 6 billion if you think of the diploid. They have the same 25,000 genes. What's different? It's just the expression of the genes. Which ones are on, which ones are off. What about a normal breast cell versus a breast tumor? Gene expression. What about a normal, like a young breast cell versus an old, 70-year-old versus a 30-year-old's breast? It's just gene expression, right? And if we look at this here, all of this right here represents breast cancer. And this here represents normal wild type breast. These are various genes. And what we're looking at is a heat map demonstrating gene expression. So this color here is going to be lower expression. So if I'm looking at like this particular sector of genes, like this kind of looks like yellowish, like green. So this would be lower expression here. And then if I look at this looks like higher expression. And what I'm seeing is that associated with all of these breast cancer cells, I have higher expression of all of these genes. And with all of these normal cells, I have lower expression of all of these genes. Well, I wonder what these genes are. If we look at this one, this is epidermal cell growth factor. This is an oncogene. CADM1 is an oncogene. CCL4 is an oncogene. And so we can see it makes sense. This is going to be upregulated in the cancer cells and downregulated in the normal cells. And you know what? I haven't really checked these, but you can imagine these are tumor suppressor, in which case it's downregulated there and upregulated right there. So this is just a way that you could quantify and look at. Not that you guys have to really understand anything about heat maps. Cis regulation is anything that's on the same chromosome typically. So talking about enhancers, talking about promoters and whatnot. Trans factors are going to be transcription factors coming from far away, another chromosome perhaps. Typically protein mediated. Often they're going to work together though, right? I have a promoter interacting with an enhancer through lots of these transcription factors in between. Enhancers can be upstream of a promoter over here, or they can be downstream of a promoter. Heck, they can be in the middle of the gene itself in an intron as well. For a promoter, why would we expect to see a lot of AT-rich regions, right? People have heard of like a TATA box as an example. Why all these A's and T's? Well, the promoter, we have this -35 and -10, which is typically where transcription factors are going to bind like their two arms. And the first thing we have to do is unwind the DNA to allow everything and allow transcription to occur. And it's easier to unwind and sort of melt or to unanneal two strands of DNA if they're not bound as tightly. Well, remember that CG has 3 and AT has 2 hydrogen bonds. So it makes sense that AT is disproportionately concentrated in the promoter. So we are going to have our promoter and maybe our TATA box, and we're going to have a TATA box binding protein and then a couple of transcription factors and then more transcription factors, and eventually they are going to recruit the polymerase. Everything is unwound, and the polymerase starts moving to the right, to the right, to the right until it hits this +1 site, and that's where it's going to start incorporating nucleotides. Nucleotides that are going to be identical to the coding strand, but which are perfect, but are literally measured up against the template strand. So they match the coding, but they're measured up against the template. And because it's the antithesis of the template and the coding is the antithesis of the template, then that means the mRNA is actually the same as the coding. Keep in mind this is upstream and this is considered downstream typically. So what I was just saying, we can see a lot clearer here. Remember, this is 3' and this is 5', which means this would be 5' and this would be 3'. We are transcribing in the 5' to 3' direction. So here's my coding strand. We have TAC, and the mRNA we see is going to be UAC. It's identical except for that T's become U's. But we get this by, it's not ever actually even seeing the coding strand. It's literally copying the template strand, also known as the anti-strand strand, also known as the non-coding strand. So that is where we're literally laying down these new nucleotides to generate our mRNA. Transcription occurs first, and then after that, translation. If we look at something like this, we see this is our mRNA. We better be able to infer which one of these is the coding strand. Boom, it's the one that looks exactly the same as the mRNA, except for T's become U's. We have to splice out introns. So I think of this as like the opposite of the Snyder Cut, right? He had this. We had a short movie that was like 2 hours, and he said, I'm going to put in all of this extra deleted scenes and footage to make it really long. It's the exact opposite. Here we're cutting out all of the footage that no one really wants to see, and we're making it a nice, tight, condensed, final product with no extra garbage. So it's exons only. So the exons are ultimately expressed. We're going to come back to these UTRs as well in a hot second, so keep that in mind. Bacteria do not have introns, so if we want to splice an insulin gene from humans into bacteria, you have to cut out all of the introns first. Typically, you do this by transcribing it into mRNA, reverse transcribing it into cDNA, and then you can put that into the bacteria because they just don't know how to. They'll try to express the introns as if they'll try to make amino acids out of them, which will be nonsensical. So this is for introns to understand the splicing. A nucleo, is like an SN2 reaction, comes here, and then that leaves, and we get this, right? This picture actually stinks. I think this is in first aid. Technically, that should go over there, and there's a couple other things about this that are misleading. So let's just go ahead and look at this. So what we're seeing right here is this picture right here, this third one. So here I have my spliceosome. Here I have exon 1. Here I have exon 2. And we have something called a branch point. We're going to have an adenine. Nucleophilic attack. This is going to attack and cause this. So it's going to attack sort of like right here. And this is going to then be our leaving group and go right there. So again, we have our, this on its own. And again, the adenine is just attached to the phosphate. And so now this OH is going to go ahead and attack there. And then that's going to leave. And boom, we now get a lariat just like that. This is the exact same as group 2 self-splicing introns, except we do not have a spliceosome. It's the exact same thing over here. This attacks there. We get a leaving group. The thing that just left now is attacking exon 2. It's connecting to exon 2, but it's causing this to leave in the process. And again, we get a cyclical lariat. For group 1 self-splicing, we have an exogenous guanine. So an exogenous guanine floats over. It's not actually in the nucleic acid structure of the intron. It's exogenous. It's like a monomer. It just happens to be close by. So it goes over. It attacks. It just literally attaches to that. Of course, that is was a leaving group. Did leave, and then the same final step. This attacks there and causes that to go over. But because, again, it was exogenous, we do not have the cyclical lariat form. We just have this kind of linear structure right there. So there's a lot of similarities between them. There's much more similarities than there are differences. So I would just try to keep that in mind as you review the splicing. Alternative splicing is where it's, one man's intron is another man's exon is one way you could think of it. So in this case, we have 5 possible exons. Maybe one version of the protein gets all 5 exons. And that could be a protein in like a muscle cell. And you know what? Maybe in like a bone cell, for whatever reason, we don't want exon 3. And so, we have just exons 1, 2, 4, 5. And, now we don't want exon 4. We have 1, 2, 3, 5. So that would be, it'd be like we're taking, all of the footage for the Zack Snyder thing, and we're making like 10 different versions of Justice League movie based off of like which deleted scenes we wanted to keep in or not keep in. So this way you could have one gene that could potentially generate hundreds or even thousands of different versions of a protein based off of the different combinations of exons that you allow in or potentially exclude. So in addition to splicing, we're also going to add this 5' guanine cap and this poly A tail. The purpose of this is to export it from the nucleus. Getting out of the nucleus is like getting into a club, and you got to get past the bouncer, and the bouncer will only let you into the club. I guess the club now is the cytosol. We want to get out of the nucleus. You need to have the 5' cap in the poly A tail to be let out. RNA is incredibly unstable, right? Because we have this 2' OH group because it's single-stranded. This gives it extra stability because we also have exonucleases. Exonucleases, keep in mind, as an endonuclease will come in here and induce a cut. Exonucleases will try to eat up at the 5' or eat up at the 3' end. But if you have a poly A tail and you have a 5' cap, it protects you. It also helps us latch on to ribosomes for translation. All right, this is everything together. So we have our open reading frame starting with AUG. The end of the reading frame is going to be UGA or AUG or UAA, or enhancer and a promoter. So we have the +1 site, which is right here, and we're going to transcribe everything. And note that so when we cut out the introns, we still get these, we have these little black regions. So this is going to be a UTR. UTR is an untranslated region. And you might say an intron is an untranslated region, but yeah, an intron that was cut out during, right after transcription. So an untranslated region is technically something that survives the transcriptional processing, makes it out as into the mRNA, but is nevertheless not translated by the ribosome. So that is a 5' UTR and a 3' UTR. And actually, sorry, the UTRs are right here. I actually misread that. Yeah, this is just showing you the start and the stop. There we go. Cool. Bacteria transcriptional regulation. So let's say that I'm not eating milk because it's giving me bad tummy issues. So every 3 days I am ingesting milk, but that's not that often. So the bacteria in my stomach, over here, it doesn't make sense for them to, what color should I make milk? Milk will be blue, I guess. Blue milk, like Star Wars. So if I'm not making milk, if I'm not consuming milk, the bacteria in my stomach don't want to waste time and energy generating the enzymes necessary to digest milk. It's only when I ingest milk and it comes down to them that they then decide to activate the production of the machinery necessary to process milk. And so let's take a look and see exactly how that happens. Here, so this is going to be far away. This is far away. We have an LAC inhibitor gene far away on the genome. And then from here to here, we just have our whole LAC operon, which includes things like our promoter. It includes things like our operator, which can be switched on and off by the repressor. And then we have a couple of genes which are bundled together. This is a feature that bacteria have that humans don't have. We're monocistronic, one gene, one protein. They're polycistronic. Several genes can be transcribed sort of at the same time as part of like one unit. Typically, they're going to have they're going to be functionally related. So far, far away in a galaxy long, long ago, we have an inhibitor made right here. And if we just think like third graders with the shape of blocks, this shape could perfectly fit right there. So this is the natural state of affairs. An inhibitor is made that perfectly fits into the operator. It binds, and it's now a giant roadblock. We now have this, giant roadblock thing. And you know what? My polymerase here cannot get past this roadblock, this big clunky repressor, which is plugged into our operator. And so what happens? I do not get transcription of LAC-Z, beta-galactosidase needed to digest milk. LAC-Y, which permease lets the lactose in. And the transacetylase, no one really, I think, fully gets what that does, so we'll ignore that one. But this makes sense, right? We just think economically, would it make sense for the bacteria to try and make the enzymes to digest milk if I don't have any milk? No. So this is, and I'm not normally, right? We said I only have milk every couple of days. So this should be the normal state of affairs. All of this stuff should naturally opt. This should make sense. Now, I have like mocha frappuccino and there's lots of dairy in there. What's going to happen? I'm going to have milk. It's going to come in. It's going to go through my GI. It's going to go to the bacteria. Let's just think milk is lactose and lactose is allolactose. So this little milk particle right here is going to come in, and the repressor has been made so that it perfectly fits one of these little milk molecules. And what happens, just again, we're third graders with blocks, what happens when a milk molecule binds to this repressor? It changes the shape. So now does this shape, I wish I can I actually undo this? Whoa, we can't undo it. Great, so we're third graders again. Does this shape right here, does this fit into there? And the answer is no. No, it does not. What we've done is we now inactivated the repressor. And if I cannot, if I can't repress this, then what's going to happen? What's going to happen is we're now going to have our polymerase is open for business. There's no roadblock. It's going to transcribe this, and this. We're going to get all of these three enzymes. What are they going to do? They're going to help us digest milk, which makes sense because I have milk. You can only digest milk when milk is there, repressing the repressor, allowing transcription of the things necessary to digest milk. This will go, it will chew up all of the milk. It will convert the repressor back to its normal state, which makes sense because we just consumed all of the milk. We want this to go back in. This is a smart system if you think about it. So that's bacteria being like lazy or smart or economical. But we could also say that they're picky. And you could also actually, so weave this into the economics like metaphor. The idea is that they will only digest milk if there's milk there. But at the same time, they would prefer glucose. So if there is milk and lactose, they will just go for the milk, and they will probably not make much of the enzymes to digest lactose. Now, how does this manifest? Glucose is going to indirectly correlate with something called CAMP. So if you have low glucose, you have high CAMP. If you have high glucose, you have low CAMP. And cyclic AMP is needed to activate the CAP transcription factor. So if we look at this, we have a high lactose. High lactose simply means that the lactose bound to the repressor inhibited it. It doesn't bind to the operator, so the operator is free. Transcription can theoretically occur. It doesn't mean it will occur at a high rate. High glucose is there too, though. Let's just think about the economics. Do we really even want to waste time making all of this stuff that I just lactose if there's glucose there? No. And the way that this physically works is that high glucose correlates with low cyclic AMP, which means the CAP will stay inactive. You do not have this transcription factor. You will not get much transcription. On the other hand, if we look at this, high lactose, the operator is free. Low glucose means high CAMP, which means CAP is active. We can have transcription, and it will happen quite quickly because of the CAP protein. So 4 possibilities here. If we have, just think about the economics, and then we can think about what's actually going on with the molecules. Low glucose means high CAMP, means CAP is active. Lactose is available means the repressor is bound, and so it's not doing its thing, which means the operator is free, which means transcription can occur. This is the scenario where you have a lot of the lac genes expressed to digest the milk because there's milk and no glucose. High glucose and lactose. This is a double no. We can just go for the glucose, and it wouldn't even make sense to make the lactase enzymes because there's no lactose. So, nope. Low glucose and lactose is unavailable. Well, yeah, there's no glucose, so it would be nice to digest milk if we had milk, but we don't even have milk, so the repressor is going to be bound really tightly, and we're not going to waste ATP and time transcribing this particular operon, so it's not expressed. High glucose and high lactose, this is where the bacteria can afford to be picky, and they go, yeah, I know that theoretically we could transcribe all the lactase stuff because the operator is open, but you know what, we just rather go ahead and digest glucose. So the low CAMP levels means that CAP is inactive, so you'll have a very low level of expression there. Into a little summary, go ahead and take a mental snapshot or just stare at it forever. All right, so RNA is very unstable. We said that single-stranded 2' OH group. So we have a couple different kinds. rRNA is actually 85% of it. I would definitely know this. So the most ubiquitous type of RNA is going to be the ribosomal RNA. It's made in the nucleolus. tRNA is 10%, and then mRNA, which really gets all of the accolades, is the least common one. Everything is 5' to 3' except for exonuclease. Type 1 for rRNA type 2 for mRNA, so this is probably the most important one to know, even though it's only 5%, just because, I don't know, it's important. Amanitin it's from a fungus. It's an inhibitor of the type 2 RNA polymerase. And then type 3 is for TNA, but it actually, it does make, not all of the rRNA is made by type 1. A little bit, the 5S rRNA that goes into ribosomes is by type 3. Rifampin, something we use for tuberculosis, is an antibiotic. It's going to inhibit transcription. And then actinomycin can affect both eukaryotic and prokaryotic transcription. So this could be used, if against bacteria as an antibiotic. But if we want to stop eukaryotic transcription, we might use that if there's a lot of transcription, a lot of replication in a cancer.